This slide presentation is the recording of muscle tissue, how muscles transform chemical energy into mechanical energy, part 2 of 2. We know that muscles need huge amounts of adenosine triphosphate, and we also know that neurons require adenosine triphosphate. The process of releasing acetylcholine into the synaptic cleft by moving synaptic vesicles so their membranes fuse with the membrane of the synaptic end bulb requires energy in the form of adenosine triphosphate. In order for a neuron to be excitable, we have to maintain more sodium on the outside and more potassium on the inside. And in order to do that, we have to pump those ions against their concentration gradients with the help of adenosine triphosphate. So those sodium-potassium pumps for neurons as well as for skeletal muscle is important. Those require adenosine triphosphate. To move calcium back into the sarcoplasmic reticulum also requires a pump which uses adenosine triphosphate. And lastly, we know that detaching the myosin head from the myosin binding site on actin requires fresh adenosine triphosphate. Energy is normally stored in our muscles as glycogen. Remember, glycogen is stored in muscles and liver. The glycogen is further broken down by way of hydrolysis into glucose. Remember, the term hydrolysis means breaking or bursting apart with the help of water. Glucose can be further broken down in two ways. One is by anaerobic respiration, and the other is by aerobic respiration. So we need to be able to break down glucose to produce more adenosine triphosphate. Our muscles have a lot of mitochondria located inside the cells, inside the sarcoplasm. We know that mitochondria are the site of production for the chemical energy required for our bodies called adenosine triphosphate. We know that adenosine triphosphate has a lot of energy because the third phosphate group in adenosine triphosphate is held on by a very high energy bond, which when it breaks, releases energy to do work. When our body is at rest, our bloodstream will deliver fatty acids and ketone bodies to the mitochondria, which in the presence of oxygen will go through a series of chemical reactions known as the citric acid cycle or the Krebs cycle to produce adenosine triphosphate and byproducts of carbon dioxide and water. We get a lot of adenosine triphosphate when we use the mitochondria and oxygen to produce adenosine triphosphate. We get 36 to 38 molecules with aerobic respiration. 36 to 38 molecules of adenosine triphosphate are made with the process of aerobic respiration. Our bodies do not store adenosine triphosphate. We can store a tiny little bit. However, most of it has to be used, and a lot of it does get used. We have many non-contractile energy requirements in our cells, which include the sodium-potassium pump, active transport in the form of endocytosis, exocytosis. There are other pumps like the calcium pump in the sarcoplasmic reticulum. These are just some of the non-contractile processes that occur within our cells in our body that require ATP. This means even at rest, our bodies are continuously producing energy so our cells can function. When adenosine triphosphate is used for energy, remember, it's going to break down into adenosine diphosphate and inorganic phosphate. We can't have inorganic phosphate floating around on its own. However, with the help of an enzyme called creatine kinase, we can attach that inorganic phosphate to a molecule of creatine, creating creatine phosphate. And the neat thing about creatine phosphate is that the phosphate group can be taken off of that, again with the help of creatine kinase, to phosphorylate adenosine diphosphate, turning it back into adenosine triphosphate. When muscles are contracting, we look at the picture on the right-hand side. Now we're going to need more energy, and the glycogen granules that are stored in muscle tissue will start to go through a process of hydrolysis, which breaks them down into their individual components of glucose molecules. Glucose will then be further broken down by means of a process known as glycolysis. Glycolysis is anaerobic metabolism. There is no oxygen involved in glycolysis. Glycolysis breaks glucose down to form only 2 molecules of ATP and pyruvic acid. The pyruvic acid will actually go to the mitochondria, and in the presence or with the presence of oxygen, the pyruvic acid will cycle through a series of chemical reactions called the Krebs cycle or the citric acid cycle to form adenosine triphosphate and the byproducts of carbon dioxide and water. Remember, anytime we use aerobic respiration to produce ATP, we get a lot more bang for the buck. 36 to 38 molecules of adenosine triphosphate with aerobic respiration versus 2 molecules of adenosine triphosphate with glycolysis, which is considered anaerobic because there's no oxygen required. Now, as ATP is used up, and if an oxygen deficit is created because we're using more energy than we can breathe in oxygen to maintain oxygen for the mitochondria, then the body will start to produce, and by the body I mean the muscle cells, muscle fibers, will start to produce lactic acid and very small amounts of adenosine triphosphate. Now, lactic acid can be reconverted into pyruvic acid. It does take a small amount of time, but it can be reconverted. However, without oxygen, if we have too much of an oxygen debt, we won't be able to produce as much adenosine triphosphate if we don't have enough oxygen for the mitochondria to produce those large amounts of adenosine triphosphate. And here again, we're seeing the regeneration of adenosine triphosphate when we break it down into adenosine diphosphate and inorganic phosphate, and we can take the phosphate that's stored on creatine phosphate with creatine kinase and move it back onto the adenosine diphosphate to make new adenosine triphosphate. This is just another anaerobic pathway not a very powerful pathway for producing adenosine triphosphate. It does exist, which is why I'm showing it to you on this picture. Creatine is actually produced in the liver, the kidneys, and the pancreas. And because our muscles, even when they're relaxed, are going to produce excess adenosine triphosphate, most of that is going to be broken down and stored as creatine phosphate. Creatine kinase is the enzyme responsible for transferring phosphate to the creatine molecule from the adenosine triphosphate and also to the adenosine diphosphate from the creatine phosphate when the body needs more adenosine triphosphate for muscle contraction. So when contraction begins, the creatine kinase is going to take the phosphate group from the creatine phosphate and stick it back on the ADP, the adenosine diphosphate, in order to make more adenosine triphosphate. In fact, creatine phosphate is the first source of energy for muscle contraction when contraction begins. It actually provides the first 15 to 20 seconds of maximal contraction. The prefix "an" means without. Anaerobic literally means without oxygen. Anaerobic respiration does not use oxygen to produce energy. Creatine phosphate is our first source of energy. Remember, it's the first 15 to 20 seconds of maximal contraction, no oxygen involved. The second source of energy for contraction is going to come from glycolysis, where we break down glucose to make adenosine triphosphate. And breaking down glucose forms 2 pyruvic acids and 2 adenosine triphosphate molecules from 1 molecule of glucose. Now, this makes the body slightly acidic. However, it's nothing that is going to take a person beyond the boundaries of function. This is good for very short bursts of strength where we have 30 to 40 seconds of maximal contraction. In other words, we start with creatine phosphate, first 15 to 20, no oxygen required, totally anaerobic, move into glycolysis, 30 to 40 seconds of maximal contraction, second source of energy contraction, no oxygen, anaerobic. Then we get to aerobic respiration, and sometimes this is also referred to as cellular respiration. This requires oxygen. It's used when people are resting or exercising lightly to moderately. The process of aerobic respiration includes a cycle of chemical reactions that's going to take longer. Therefore, aerobic respiration is going to be slower, but it's a lot more efficient in producing ATP. From 1 molecule of glucose, we get 2 pyruvic acids that enter the mitochondria and a yield of 36 to 38 molecules of adenosine triphosphate. Some sources say 32 molecules of adenosine triphosphate. That's neither here nor there. There's a lot more compared to anaerobic respiration. We see the byproducts listed here on the slide of carbon dioxide, water, and heat. Heat is also a byproduct of aerobic respiration, and this provides our bodies with long-term contraction. Remember that myoglobin is the protein molecule that can help store oxygen. It's similar to hemoglobin in the bloodstream, only myoglobin, the prefix myo means muscle. And glycogen is stored in granules in muscle cells, and that's where we get our oxygen and our energy for aerobic respiration. Looking at these comparisons of aerobic versus anaerobic, you can see that they can both use glucose and they generate energy. The biggest thing I want you to see is that with anaerobic, it's inefficient. We get 2 molecules of adenosine triphosphate. With aerobic, we are much more efficient, 36 to 38 molecules of adenosine triphosphate. We must have oxygen with aerobic respiration, and we have no oxygen with anaerobic respiration. Know that with aerobic respiration, we have the production of ATP with oxygen in the mitochondria. With anaerobic respiration, we've got 2 forms, including the use of creatine phosphate and glycolysis as the 2 mechanisms to produce adenosine triphosphate. If you've ever watched a person run a marathon and just stop at a certain point and breathe, that individual has reached the point of what is known as oxygen debt. The individual simply is not able to get enough oxygen into a system to supply the demands for the mitochondria to produce adenosine triphosphate. And oxygen debt, when a person reaches that point, the body starts producing lactic acid anaerobically. Oxygen debt is defined as the oxygen needed to get rid of the lactic acid. This can be done in the liver with the presence of oxygen, where the lactic acid and oxygen are going to go through chemical reactions, creating carbon dioxide and water. Any persistent lactic acid can be converted further by the liver back into glucose. And during this time, we're going to see that oxygen is replenished on the myoglobin, and creatine phosphate and adenosine triphosphate can be resynthesized. During oxygen debt or oxygen recovery uptake, you may see the person breathing deeply and heavily and have a rapid rate of respiration. This process may last minutes to hours. And again, the extra oxygen that the person is taking in once he or she stops is basically used for restoring metabolic conditions. We discussed earlier the fact that there is a special protein called dystrophin that anchors the myofibrils to the sarcolemma of the muscle fiber. Imagine my arm in my sleeve as a myofibril and the sleeve being the sarcolemma. If there's no anchor, then my myofibrils may contract, but the sarcolemma doesn't contract because it's not anchored properly. If the dystrophin anchors the myofibril to the sarcolemma, then when the myofibril shortens, the sarcolemma shortens as well. So imagine Duchenne's muscular dystrophy as the myofibrils contracting inside the skeletal muscle fiber. However, no true shortening is occurring in that muscle fiber because the dystrophin protein that normally anchors the myofibrils was just not built correctly. This is going to cause progressive weakening of the muscles and is a hereditary disorder that primarily affects males. Most do not live past their 20s. In this cross-section of a person's muscle tissue with Duchenne's muscular dystrophy, you can see that a lot of the myofibrils here have been replaced with adipose connective tissue. Because these muscle fibers, if they're not used, the proteins start to break down, and then that's why the muscle just continues to become progressively weak. And there's a lot of different presentations with a person who's got Duchenne's muscular dystrophy. You can read about this. It's a very sad progressive disease. Next, we're going to discuss isotonic versus isometric contractions. Remember the prefix iso means the same. Isotonic, same tone or tension. Isometric, same length. Isotonic contractions mean that the tension is constant, but the length changes. Isotonic contractions are used for movement and moving things. When we shorten the muscle, that is referred to as a concentric isotonic contraction. When the muscle lengthens, that is an eccentric contraction. So we have concentric muscle shortening, eccentric muscle lengthening. If there's no change in muscle length, now we have isometric contractions. So there's no change in the muscle length, and that would be an example of an isometric contraction. Usually isometric contractions are used for maintaining posture, stabilizing joints, and holding objects. Another example of an isometric contraction would be if I were to push against this wall, I'm contracting the muscles in my arms, but they're not moving, and I'm not moving the wall. That would be another example of an isometric contraction. We know in order for skeletal muscle to contract, there has got to be a nerve taking information in the form of an action potential, which you know is an electrical impulse, to the skeletal muscle fibers. We see that these motor neurons have cell bodies that reside in the anterior horn of the gray matter of the spinal cord. Even though it appears brown, the darker area of the spinal cord in the front is the anterior horn of gray matter. The cell bodies are those little balls that you see inside that area. The axons of these neurons exit the spinal cord, forming nerves that are going to go to the skeletal muscles. As they reach the end, they branch out to go to different varying numbers of skeletal muscle fibers. Remember, a motor unit is one somatic motor neuron plus all the skeletal muscle fibers it innervates. If you look at the purple motor neuron and follow it to the end, you will notice that it innervates this skeletal muscle fiber and this skeletal muscle fiber. That's two. If we look at the green motor neuron, we see that it innervates this muscle fiber, and this muscle fiber. Therefore, it innervates three muscle fibers. Precise control of motion occurs with smaller motor units. Motor units that have anywhere between 2 and 20 muscle fibers per unit are going to give us precise, fine motor control. Whereas powerful control of our large muscles and our postural muscles is going to occur because we have larger motor units. 2,000 to 3,000 fibers, muscle fibers per motor unit will be innervated with larger muscles. The strength of a contraction is going to be dependent on the size of the motor unit. In other words, how many muscle fibers does it branch out to, as well as the number of units activated all at once. I do not have to recruit many motor units to lift up a cell phone. I have to recruit more motor units to lift up this weight. Therefore, we can increase the strength of a contraction by recruiting motor units. Within the sarcomere itself, there's actually an ideal length of overlap between the myosin binding sites on actin and the myosin heads. And the ideal length for maximal tension to occur, to be generated during a contraction, is considered to be 80 to 120% of the resting length. Again, that maximizes the overlap of myosin binding sites on the actin molecules and the myosin heads for the best contraction to occur. A muscle twitch is the response of one motor unit to a single action potential. These are usually diagrammed on myograms, and you'll notice that from the time of stimulus, there is what is referred to as a latent period, which is usually the first few milliseconds that occur between the stimulus and the actual contraction. The latent period occurs because the action potential has to cause the release of calcium in the sarcoplasm for contraction to occur. The contraction period occurs during the time that cross bridges form. The relaxation period occurs when calcium is pumped back into the sarcoplasmic reticulum and cross bridges are broken with fresh adenosine triphosphate. If a muscle fiber is stimulated while a previous twitch is already occurring, then we see a phenomenon that the second contraction or twitch is going to be stronger, and that's something known as wave summation. There is a slight bit of relaxation, but not complete relaxation in wave summation. With tetanus, we see a complete contraction with no relaxation. So we have continuous contractions and no relaxation with tetanus. TREP is also known as the staircase effect, and what happens is they've noticed with twitch contractions that with complete relaxation in between each one, if they continue to stimulate the muscle to contract immediately after relaxation, they see more efficient contractions. And this actually results, the TREP effect or staircase effect, of building the strength of contraction or tension of contraction occurs because there is a higher concentration of calcium in the sarcoplasm. And this can be maintained as long as there is adequate adenosine triphosphate. In terms of summation of contractions, that's dependent on the frequency of the stimulation. If we have one stimulus, one action potential, which is an electrical current, stimulating the muscle, the result is one muscle twitch, one contraction. If we add one stimulus with another stimulus before the muscle gets to relax completely, we see wave summation. If we continue to add stimuli to the muscle, allowing slight relaxation in between, but not complete relaxation in between, we see more wave summation, which is sometimes referred to as unfused tetanus. If we see such frequent stimulation that there is no relaxation in between contractions, that is known as fused tetanus. Please know for this class, I do not need you to know the difference between unfused tetanus and fused tetanus. You can simply understand a twitch contraction, wave summation, the TREP or staircase effect, and tetanus. Muscle tone occurs because we always have a small amount of tension in resting muscles, and these are due to weak contractions of motor units. This helps keep our muscles firm even though they're not contracted, and is present in skeletal muscle. Most books say that it occurs while people are conscious because we use it for postural stability and joint stability when we're moving around and lifting things. It also includes smooth muscle to maintain the tone in our blood vessels and our organs. So it really actually occurs all the time. Hypotonia is a condition where we have an absence of muscle tone. So there's an absence of these low-level contractions, and that leads to muscle atrophy. Muscle atrophy occurs when the proteins are not being used and they start to break down, leading to a loss of muscle mass as well as muscle tone. That's what atrophy is a loss of muscle mass. Hypotonia can result from damage to the cerebellum, which is a little area just posterior to and inferior to what you know as your brain. And poliomyelitis is another condition that can cause hypotonia. Usually individuals with hypotonia have such decreased muscle tone that they appear flaccid or limp, and they will have very weak or absent reflexes. Hypertonia is the opposite. It's excessive muscle tone, contraction, and hyperreflexia, usually resulting from damage to the upper motor neurons. Remember when we looked at the motor unit, which was a single somatic motor neuron that goes from the spinal cord to the skeletal muscle? That neuron is referred to as a lower motor neuron. The signal to contract actually starts from the primary motor cortex in your brain. That's where the upper motor neurons live. These neurons have axons that travel down through the spinal cord and will communicate with the neurons in the spinal cord. So upper motor neurons in the brain, lower motor neurons in the spinal cord. Damage to the upper motor neurons usually occurs when there is some kind of hypoxia during the birthing process, where maybe the cord is wrapped around the neck and the brain can't get oxygen correctly. Cerebral palsy is a good example of upper motor neuron lesions that result from hypoxia to the brain, where it damages those upper motor neurons. And you'll often see people with cerebral palsy in a very contracted state. It's not unusual for doctors to remedy that contracted state with Botox injections, because botulism toxin actually prevents the release of acetylcholine at the myoneural junction and prevents the muscle from contracting, and that actually helps relax the state of hypertonia. Within muscles, we have different types of skeletal muscle fibers. They may be more prevalent in certain muscles than in others. For instance, we have red fibers, which are slow contracting oxidative fibers. They use oxygen. That's what oxidative means. They use oxygen. Therefore, they are going to be high in myoglobin. When oxygen attaches to myoglobin, it gives it this nice red appearance, just like when oxygen attaches to hemoglobin in your blood, gives it a nice red appearance. Red muscle fibers have abundant mitochondria and lots of capillaries, which are those tiny little blood vessels to carry blood and oxygen to these skeletal muscle fibers. Because they are oxidative, the preferred method of energy production is going to be cellular respiration, which is aerobic respiration because we use oxygen. And these types of fibers are important for people who do endurance exercises like long distance running, triathlons. And they tend to have lean muscles with very big blood vessels to help carry oxygen and nutrients to those skeletal muscles. White fibers are fast glycolytic fibers. Glycolytic means it's going to use glycolysis, which is anaerobic, for its preferred method of producing energy. So white fibers have a white appearance because they are low in myoglobin. They don't have very many mitochondria because they're not using aerobic respiration. They have fewer capillaries because, again, they're not using oxygen as much. These white fibers are going to be more prevalent in people who use their muscles for short strength exercises. The muscles tend to be bulkier, so weightlifters are going to have more white skeletal muscle fibers. Now, as far as red fibers go, postural fibers are always going to have more of the slow oxidative fibers within them. If you've ever eaten a chicken, the legs and the thighs are the dark meat, and there's a little bit of dark meat on the back of the chicken. That's the postural part for the chicken that holds it up in the back so it can stand on its legs. So the legs and the back are dark meat because those are the postural muscles for the chicken. Again, they're dark because of the myoglobin. Now we have different kinds of fibers. There are intermediate fibers as well. And all of these different types of fibers have different functions. Again, the red fibers are going to be slow oxidative using aerobic means for producing adenosine triphosphate, whereas the white fibers are fast glycolytic fibers, which are lower in myoglobin, low in mitochondria, and have fewer capillaries because they're doing production of adenosine triphosphate anaerobically. When muscle is used more often than what we would consider normal for that individual, we may see hypertrophy. And hypertrophy can occur not just because a person is using his muscles more. There are ways to artificially make muscles grow bigger, which include the use of growth hormones and anabolic steroids. With hypertrophy, what happens is we see an increase in the number of myofibrils in the existing cells, in the existing muscle fibers. That's what causes muscles to grow bigger. And motor neurons always need to be intact for muscles to work correctly. We either use it or we lose it. With cardiac muscle tissue, we mentioned before that the cells are branched. And here we have two pictures where you can obviously see the branching in the cardiac muscle tissue that we couldn't see readily previously. We may see one or two centrally located nuclei with cardiac muscle tissue. We have striations in cardiac muscle tissue, which is easy to see even on this picture of the actual slide below. You can see those striations. That's due to the presence of the sarcomeres, which are the functional contracting units produced by the overlapping actin and myosin. We know that cardiac muscle tissue also contains these special structures referred to as intercalated discs. Intercalated discs have desmosomes, which are like spot welds or Velcro, and the desmosomes hold the cardiac muscle tissue together during very forceful contraction. The intercalated discs also have gap junctions, which allow ions to flow from one muscle fiber to the next to allow for the muscle tissue to contract as a unit. There are 4 chambers in the heart. The 2 upper chambers are the atria. The 2 lower chambers are the ventricles. When the heart beats, it beats like this. Atria, ventricles, pause. Atria, ventricles, pause. Atria, ventricles, pause. When the atria beat together, they beat together in what is referred to as a functional syncytium. The word syncytium has the beginning S-Y-N-C, sync. They beat in sync as a functional syncytium. It's actually S-Y-N-C-I-T-I-U-M. That's the complete word, syncytium. We see the striations in in cardiac muscle tissue because of the presence of sarcomeres, because the heart has to beat 24 hours a day, 7 days a week. It has many mitochondria, and calcium is stored not only in sarcoplasmic reticulum, it also comes from the interstitial fluid surrounding cardiac muscle cells. Unlike skeletal muscle tissue, which is voluntary, cardiac muscle tissue is involuntary. It is also autorhythmic. It has cells that will cause it to beat on its own. Cardiac muscle tissue can contract 10 to 15 times longer than skeletal muscle because it is exposed to calcium longer. It has to have oxygen at all times and can also use lactic and fatty acids for energy because it never stops. Cardiac muscle cannot reach tetany because it has a longer refractory period and also cannot be stimulated until it has relaxed. When cardiac muscle tissue is damaged, it cannot regenerate. Damage leads to collagen scars. Smooth muscle tissue is also involuntary. The cells of smooth muscle tissue are tapered on the ends and thicker in the middle. They have one centrally located nucleus. There are no sarcomeres inside these cells, therefore you will not see striations. They have a very scanty, poorly deformed sarcoplasmic reticulum, and the fibers actually twist when they contract. It's kind of like wringing out a towel. You'll notice that there are 2 types of smooth muscle tissue on this picture. We have visceral or single unit smooth muscle and multi-unit smooth muscle. Pay attention to the fact that special neurons innervate the smooth muscle cells. These neurons are called autonomic neurons. When I think of the word autonomic, I think automatic because it sends signals automatically. I don't have to consciously think about it. There's something else you should pay attention to in this picture. Notice that the multi-unit smooth muscle has each cell with its own branch of these autonomic neurons, whereas the visceral or single unit smooth muscle does not. That might help you remember which is which. It might help you make that distinction. Visceral or single unit smooth muscle is usually the type of smooth muscle that we see lining hollow organs. For instance, the intestines actually have 2 layers of smooth muscle tissue. The layer on the outside of the intestines is a layer that runs the length of your intestines. Therefore, it is the longitudinal layer of smooth muscle. And then on the inner portion, we see a circular layer of smooth muscle tissue. When the intestines contract, they contract in a wave known as peristalsis. And basically what happens is the longitudinal layer shortens and the circular layer squeezes. So it kind of goes squeeze, shorten, squeeze, shorten, squeeze, shorten, and that pushes the food that you're digesting in your intestines, which by that point has turned into something called a bolus, through your intestines. Again, on this slide, we see smooth muscle is involuntary. There are 2 types, visceral single unit or multi-unit. Visceral single unit is the type of smooth muscle found in tubular or hollow organs like the uterus, the urinary bladder, the stomach, the intestines. It is autorhythmic and has gap junctions. The gap junctions allow waves of contraction to occur. Multi-unit smooth muscle is named so because each fiber has its own motor unit. Remember the picture I showed you previously where you saw the individual smooth muscle cells and each one had a branch of the autonomic nerve going to them? Multi-unit smooth muscle is found in large arteries, the bronchial tree. It is the type of smooth muscle that makes up the erector pili muscles, the iris of your eye, which is the colored portion of your eye, whether you have brown eyes or blue eyes, that's actually multi-unit smooth muscle, as well as the ciliary muscle, which is the muscle that moves the lens of your eye to change shape of the lens to allow you to accommodate seeing far away or close up. Smooth muscle does regenerate better than both skeletal muscle, and we know cardiac muscle doesn't regenerate really, it just forms scar tissue. However, smooth muscle can regenerate fairly well. Smooth muscle tissue exhibits a really interesting phenomenon known as the stretch relaxation response. And basically what that means is that it can stretch and then relax at that new distended or stretched shape. And smooth muscle is actually capable of stretching or distending to a great deal, which is very obvious when we see someone who is pregnant. The uterus is comprised of smooth muscle tissue, and as that developing baby grows and grows, the uterus will continue to stretch and stretch to accommodate that growing baby. The other example of a stretch relaxation response is evident with overeating. Normally, a stomach is the size or same size as someone's fist. If you've ever gone to the Marietta Diner, you may have noticed that a person might receive a plate that's probably got portions that are 4 to 5 times larger than that individual's fist. And the stomach can actually stretch and distend and stay stretched, and people don't feel full when the stomach stretches that much, and that's why lap band surgeries became very popular. Now, if you look at the picture on the left-hand side of this screen, you'll notice a much more detailed picture of the smooth muscle fibers. And smooth muscle fibers don't have striations because they don't have sarcomeres. And they do have overlapping thick and thin filaments, but the myosin filaments are a lot shorter than the actin filaments, and the myosin, the type of myosin, actually differs from the myosin found in skeletal muscle. And the ratio of thick to thin filaments is much lower in smooth muscle than it is in skeletal muscle. So when smooth muscle gets an impulse to contract, there's actually no troponin complex in smooth muscle the way there is in skeletal muscle. Instead, there's a protein called calmodulin that acts as a calcium binding site. And you'll notice in this picture on the left that there are intermediate filaments in smooth muscle, and these are basically a lattice-like arrangement of non-contractile filaments that resist tension. And they're attached at regular intervals to structures in the cytoplasm called dense bodies. The dense bodies are actually anchored to the sarcolemma. Now you can see the thick myosin and thin actin filaments within this smooth muscle fiber. They're actually arranged diagonally. And there are bundles of these little contractile proteins that kind of crisscross within the smooth muscle cell, so they spiral down its long axis, kind of like the stripes on a candy cane. And that way, when they contract, because they're anchored to the dense bodies, they actually pull on the dense bodies, and that kind of causes that twisting motion where the sarcolemma bulges outward like, again, the ringing of a sponge. That's the best way I could describe that contraction. I'm sorry, I realized I wasn't in view this whole time. At any rate, that's the end of this particular chapter. I'm going to start recording the next chapter, and I hope that this helped make a little bit more sense out of muscle tissue.